Okay, let's start the class now. So, hello everyone. Thank you all for joining. So, today is the 11th problem solving session for the course Environmental Chemistry and Microbiology. Uh, the course instructors are Professor Anjali Pal and Professor Sudha Goel from IIT Kharagpur, and I'm Shrija Ghosh, a PMRF from IIT Kanpur. Uh, Today, we'll discuss the questions related to your 11th week's assignment. So, I've prepared certain questions which are related to the questions given to you, and we'll discuss all the questions. And if you have any doubt in any of the questions, or if you have any doubt in any of the topics of this week, please let me know. I'll try to help you out. Uh, so I'll be asking the questions, and all of you are requested to write the correct answers in the chat box. If you know the answer, please write the answers, and then we can have a discussion if the answer you gave is right or wrong. Okay. So shall I start the class? Okay. Let's start with the first question. Question number 1. Ringworm is a disease caused by, options are A. Fungus, B. Bacteria, C. Tapeworm, and D. Roundworm. Ringworm is a disease that is caused by fungus, bacteria, tapeworm, or roundworm. All of you write the answers in the chat box. What do you think is the correct answer? Which of the following causes ringworm? Okay. I can see most of you have answered in the chat box. Option A, that is fungus. Let's see the correct answer. Fungus is the correct answer. So, ringworm is a skin disease that is caused by fungus. It is also known as dermatophytosis or dermatophyte infection. It's a fungal infection of the skin. So, you all must have seen the ad of ring guard cream. So, the circular skin lesion that they show, that is actually the ringworm. Ringworm infection that can affect both humans and animals. Infection initially appears as a red patch on the affected area of the skin, later may spread to other parts of the body. It may affect the scalp, feet, nail, groin, beard, and other areas. Okay, so it is a itchy patch that occurs on the skin, and it is caused by a fungus. Okay, so it's a fungal infection of the skin. I hope that's clear. If anyone has any question regarding any of the topics that are discussed today, please write your doubts in the chat box, or you can unmute and ask whatever is comfortable. Okay, moving on to the next question. Question number 2. Teardrops are rich in, option A amylase, option B phosphate, option C lysozyme, and option D protease. Teardrops are rich in amylase, phosphate, lysozyme, protease. What would be the correct answer? All of you write the answers in the chat box. Okay, I can see 6 of you have answered option C. Option C, that is lysozyme. Let's see if that is the correct answer or not. That is the correct answer. So teardrops are rich in lysozyme. Lysozyme is the enzyme found in the secretions of the lacrimal gland, that is tears, of animals, and in nasal mucus, gastric secretion, and egg white. So these are the other 3 places where you can find lysozyme other than tears. So nasal mucus, gastric secretion, and egg whites. Okay. This and lysozyme was discovered in 1921 by Sir Alexander Fleming, and it catalyzes the breakdown of certain carbohydrate found in cell wall of certain bacteria. So bacterial cell wall is broken down by these enzymes. So these enzymes are kind of defensive. Uh, they provide a defensive mechanism towards different bacteria. So since these enzymes can break down the cell wall of these bacteria, and once the cell wall is broken, the cell eventually dies off. Okay. So presence of lysozyme in our lacrimal gland or nasal mucus, it the presence is actually sign of defense mechanism against different pathogenic bacteria, so that any bacteria tries to enter our body, these enzymes can break down the cell wall and cause the cell death of the bacteria. Okay, is that clear? If you have any question, please let me know. Okay, moving on to the next question. What is the importance of epidermis regarding human immunity to microbes? What is the importance of epidermis regarding human immunity to microbes? It is the first layer of skin which helps in defending the human body from microbes. Option B, epidermis keeps the body temperature sufficient to kill the microbes. Option C, there is no specific role of epidermis in preventing disease. And option D, it is the last layer of the skin which helps in defending microbes from reaching the human body. What is the correct answer? What's the importance of epidermis? All of you write your answers in the chat box. What is the importance of epidermis? I can see only 4 answers in the chat box. Anyone else wants to answer? Okay. So most of you have answered and all of you are agreeing with option A. It is the first layer of skin which helps in defending the human body from microbes. That is the correct answer. So epidermis is the topmost layer of the skin. So human skin has 3 layers. First outermost is the epidermis, then comes the dermis, and then hypodermis. Okay. So epidermis is like the outer covering of our body. Okay, so imagine it like the if you are buying any food item from market, you get it in a plastic wrapper. Okay, outside covering is there, right? So imagine epidermis like that outside covering of your body. So whenever we are buying any food item and it is wrapped in a packet, we feel that it is more hygienic because it is not in contact with the surrounding environment than a open uh, some food item that is just left outside and open without any cover or package, right? So the reason why it is safe is because it is not in contact with the outer atmosphere, and in turn it is not in contact with any germs. Similarly, our body needs protection from the microbes and germs that are around in the air. So for that, epidermis acts like a outer covering for our body which protects us from uh, getting in contact with the outer air or outer uh, atmosphere or the microbes present in the air directly to our organs and directly to our body. Okay, so epidermis acts as the first line of defense basically. Okay, I hope that's clear. It's the first security that a microbe has to pass through before entering our body. That's why if you get any cuts or wounds, it is advised to put some antiseptic lotion that can prevent the microbial growth, because now your epidermis is broken. So any microbe can enter through that wound. So if you want to prevent any microbial infection, it is better to put some antiseptic chemical that can stop or inhibit the microbial growth, so that no bacteria or no microorganism can enter your body and cause some kind of disease or infection. Okay, so epidermis acts like a protective layer for your body, and if broken, you need to put some other chemical agent to prevent any microorganism to enter. Okay, I hope that's clear. I'll move to the next question. Question number 4. What causes bubonic plague? Which of the following causes bubonic plague? Option A, Streptococcus mutans. Option B, Porphyromonas species. Option C, Prevotella intermedia. Option D, Yersinia pestis. Which of the following causes bubonic plague? Streptococcus mutans, Porphyromonas species, Prevotella intermedia, and Yersinia pestis. What would be the correct answer? All of you write your answers in the chat box. Okay, I can see 5 of you have answered option D, Yersinia pestis. Let's see the correct answer. That is the correct answer. So, bubonic plague is caused by Yersinia pestis. So, Yersinia pestis is a bacterium. It's one of the plague bacterium, and there are three types of plague. So, bubonic plague is one of those three types of plague, and it is caused by Yersinia pestis. And it is an infection that is spread mostly to humans by infected fleas that travel on rodents. So there are fleas are like insects, small insects. So this is a picture of that flea. So oriental rat flea, that the scientific name is Xenosylla uh, chiopus, and this is the flea that is infected by the bacterium Yersinia pestis. So, Yersinia pestis resides in the gut of these flea. So, you can see the dark mass in the gut here. So, that is the bacterium, and the foregut of this flea is blocked by Yersinia pestis biofilm. When the flea attempts to feed on an uninfected host, Yersinia pestis from the foregut is regurgitated to the wound causing infection. So basically, these insects, they bite the rodents, different rodents like mouse, rats, and then those rodents will spread these bacterium to humans. So there are two levels of vector transmission before the bacterium is reaching to humans. So the fleas will infect the rodents, and then rodents will infect the humans. Okay, and how are these flea infects? So these flea have the bacteria in their gut, and in their gut these bacteria form a biofilm. What is a biofilm? So biofilm is also kind of a defense mechanism that is acquired by the bacteria to survive in the environment or in the host and cause diseases. So what happens is, big large number of bacteria, they come together and form a film like structure by attaching to each other by some polysaccharide or some kind of material, okay, and they form like a film. Now that film is strong enough that cannot be penetrated easily by different drugs. So when I'm applying a drug, what is happening, the drug will go and enter the bacterial cell and cause the cell lysis, or cause some kind of cell developmental issue that will eventually lead to cell death. Okay, so either it can block the protein synthesis, or it can block the cell wall formation, or it can break the cell wall, whatever is the mechanism of that particular drug. Now what happens when we are when the bacteria are making a biofilm, and then we are administrating the drug now cannot enter the biofilm. The biofilm is basically a collection of bacteria that has made a film, and now the drug cannot enter that film. So all the bacteria are kind of resistant to that drug because the drug cannot kill the bacteria. So that is a defense mechanism that is acquired by the bacteria to survive. Okay, so now in these flea, in the gut of these flea, these Yersinia pestis bacteria, they form a biofilm, and then when the flea bites the rodent, the bacteria will get transmitted to the rodent from the gut through the bite mark wherever it is biting, and then from the rodent it can spread to humans and it can cause plague. I hope that's clear. Anybody has any question or any doubt? Okay, moving on to the next question. Proteus species affect dash tissues and cause dash diseases. Proteus species affect dash tissue and cause dash disease. So options are: option A, tissue is throat epithelium and disease is pyelonephritis. Option B, tissue is small intestine epithelium, disease is cholera. Option C, tissue is kidney medulla, disease is pyelonephritis. And option D, tissue is kidney medulla and disease is spontaneous abortion in cattle. Proteus species affect dash tissue and cause dash disease. Which tissue? Mopia, what do you want me to explain again? The previous question. Which part do you have a doubt in? Can you elaborate? How the bacteria affects. So so you understood the biofilm formation, the concept behind biofilm formation, or that is the part you have a doubt in? Okay, so basically now the bacteria is forming a biofilm inside the gut of these insects. Okay, so when the insect is biting a rodent, let's say the insect goes and bites a rat. Okay, so wherever it is biting the rat, from in that point, now when the insect is biting, basically the saliva will be in contact with the bite part of the rat, right? So what will happen is these bacteria, Yersinia pestis, they will travel from the gut of these insects, these flea, and enter the rat's body through the bite. Okay, so like how mosquito bites you, you get a inflammation there, you get a redness there, right? So similarly, when any other insect also, when it bites us, we get some kind of inflammation and there's a bite mark. From that bite mark, uh, something can be transmitted, like how malarial parasite or dengue virus is transmitted through mosquito bite in humans. Similarly, these bacterium, Yersinia pestis, they can enter the rat's body through these flea, because these flea are carrying these bacteria in their gut, in their stomach, and then from the gut it will come out through the mouth and enter the rat's body, and then from the rodents it can enter humans and cause the plague in humans. Is that clear? Okay. For this question, I can see some of you have written the answer already. Anyone else wants to write the answer? Okay. So three of you have answered for this question. They were answered option C. So Proteus species affect kidney medulla and cause pyelonephritis disease. That is the correct answer. So option C is the correct answer. So Proteus species, they can infect the kidney medulla. So this is a picture of a healthy kidney, and this is a picture of a inflamed kidney which is infected by these Proteus bacteria. So the medulla region of the kidney gets infected, and the infection is named as pyelonephritis. So the term nephritis is generally associated with the kidney. So pyelonephritis is the infection, and it is a type of urinary tract infection that generally begins in the urethra or bladder and travels to one or both the kidneys. And when this infection reaches the kidney, it can inflame the medulla. You can see the inflammation, the scarring, and uh, when once the infection progress, there is pus formation that happens, and pus is filled in the medulla of the kidney. Okay, that's clear.
Ma'am, could you explain it one again?
Yeah. So Proteus is a big type of bacteria. Okay. So and generally they cause a urinary tract infection. So first the bacteria will infect the urethra, from where your urine passes on. So from the urethra it can eventually travel to the kidneys. Okay. So if it is not treated at an early stage, it can reach the kidneys. Now you have two kidneys, so it can either infect one or both the kidneys, and it actually causes inflammation in the medulla region of the kidney. So this is how a healthy kidney will look. And once this bacteria infects these kidney, you will have certain inflammation, scarring in the medulla, and eventually pus gets filled in the kidney, and that pus will basically that pus will have these bacterial uh, cells. Okay. And eventually your kidney will stop functioning properly. Your function of the kidney is to filter things out, right? So that function won't happen properly. So Proteus bacteria, they causes urinary tract infection, which eventually leads to kidney infection, and that kidney infection is called pyelonephritis, and it infects the medulla region of the kidney. Okay. So
Okay.
Proteus
Thank you,
species,
ma'am.
yeah, they infect kidney medulla and causes pyelonephritis disease. Moving to the next question, which among the following is not a host to host epidemic? Which among the following is not a host to host epidemic? Options are option A H1N1, option B hepatitis, option C influenza, and option D COVID-19. Which among the following is not a host to host epidemic? Okay. Write your answers in the chat box. What do you mean by host to host epidemic? Something that can transfer from one host to another directly. Okay. So, a disease that can be transmitted from me to you directly. That means it's a host to host epidemic. Which of the following is not a host to host epidemic? All of you write your answers. Okay, I can see three answers in the chat box. Anyone else wants to write the answer? Let's see the correct answers. Three of you have answered option B, hepatitis. Let's see. Hepatitis is the correct answer. It is not a host to host transmitted diseases. All the other diseases here, which are caused by H1N1 virus, influenza virus, and COVID 19 virus, you all know those are host to host transmittable diseases. So, for COVID 19, you all know that it was being transferred from one person to another directly. If I have the infection and if you are in direct contact with me, you can also get the infection from me through my basically, if I'm sneezing, the aerosols will be there, and then through any form of contact, these viruses can spread. Same goes for H1N1 and influenza. Okay, influenza is the virus that causes common cold. So you all are aware of influenza virus, COVID 19 virus, H1N1 virus. Hepatitis, on the other hand, it cannot be directly transferred from one host to another. Hepatitis A is an inflammation of the liver caused by hepatitis A virus. These viruses are primarily spread when an uninfected or unvaccinated person ingests food or water that is contaminated with feces of an infected person. So, if a person is infected with hepatitis, their feces will carry these viruses. Okay, and if the food or water is contaminated, contains fecal contamination, that can actually transmit the virus from one person to another. So it's not a direct transmission. The disease is closely associated with unsafe water and food, inadequate sanitation, and poor personal hygiene. So that's why uh, we are all advised to follow proper hygiene, maintain proper hygiene, uh, wash your hands before consuming any food, do not drink any water from a random source which you don't know whether it is safe to drink or not, because most of our rivers nowadays are contaminated with feces. Okay, all of them contain fecal contamination because the sewage and all are directly uh, flown into these rivers, directly discharged into these rivers. So that's why we in India, all of us, most of us have the water purifiers in our home. Those systems are required because the water we are getting directly from the natural sources are not safe for drinking, and we don't know what type of fecal contamination has. So if a person infected with hepatitis A, and that person's fecal contamination is there in your food or water, that can cause infection. So it is always advised to maintain proper hygiene, wash your hands properly, and don't drink unsafe food or water. Okay, don't drink or don't eat unsafe food or water sources. Okay, I hope that's clear to everyone. Moving to the next question, question number 7. Identify the vector of Lyme disease. Identify the vector of Lyme disease. Options are option A, Aedes mosquitoes, option B, Anopheles mosquitoes, option C, black-legged ticks, and option D, cattles. Identify the vector of Lyme disease. Aedes mosquitoes, Anopheles mosquitoes, black-legged ticks, and cattles. What would be the correct answer? All of you write your answers in the chat box. Okay. I can see many of you have answered option C, black-legged ticks. That is the correct answer. So, black-legged ticks are the vector of Lyme disease. What do you mean by vector? Vector means the insect or any organism that can carry the disease causing pathogen from one host to another. So, for Lyme disease, it is the most common vector-borne disease in United States, and it is caused by the bacterium Borrelia burgdorferi, or and rarely Borrelia mayoni. So these bacterium, these bacteria Borrelia burgdorferi, they are carried by these black-legged ticks and transmitted to humans. So humans are the primary host for these uh, bacterium, but humans are not directly infected by these bacteria. Bacteria does not enter the human body directly. They enter the human body through a vector called black-legged ticks. Again, these are small insects. These are ticks, which can bite the human, and wherever they are biting, from the site of bite, they can, and these bacteria, Borrelia burgdorferi, they can enter. Okay, so most common sign of infection is expanding red rash known as erythema migraines that appear at the site of the tick bite about a week after it occurred. So wherever it bites, it causes a red rash around the region, and that again, that is kind of a itchy, irritating rash. Okay, and that occurs because of the transmission of this bacterium, and the disease is called Lyme disease. The other three options that were there, Aedes mosquito, Anopheles mosquitoes, and cattle. So there are certain diseases that are transmitted through cattle. Aedes and Anopheles mosquitoes, they carry the famous mosquito-borne diseases, dengue and the malarial uh, pathogens. So Anopheles mosquito, female Anopheles mosquitoes can carry, sorry, female Anopheles mosquitoes can carry the malarial parasites, and same thing happens. So these parasites reside in these mosquitoes, and when the mosquito bites us directly, these parasites enter our bloodstream from the mosquito's gut. Okay, same goes for Aedes mosquitoes. So Aedes mosquito carry the dengue virus. Okay, so when these mosquitoes bite, the virus directly enters our bloodstream. Okay, so all these Aedes mosquito, Anopheles mosquito, black-legged ticks, these are all vectors of certain diseases. So black-legged tick is the vector of Lyme disease. That's clear? Okay. Moving to the next question. Identify the non-zoonotic disease from the following. Identifying the non-zoonotic disease from the following. Options are option A, anthrax, option B, rabies, option C, salmonellosis, and option D, malaria. Anthrax, rabies, salmonellosis, and malaria. Identify the non-zoonotic disease. Okay, so I can see most of you have answered option D, malaria. That is correct. So, non zoonotic disease means something that is not transmitted from animals to humans. So, zoonotic diseases, on the other hand, means the infections that are transmitted from animals to humans. Uh, so, anthrax, rabies, and salmonellosis, all of these are transferred from animals to humans, whereas malaria, it is transmitted through a mosquito, as I explained in the previous slide. So, anthrax is caused by the bacterium Bacillus anthracis, uh, and Bacillus anthracis, generally these bacteria are present in soil, and they are uh, generally they infect the animals, and from animals, people who handle these cattle and all, if they are not maintaining proper hygiene, they these bacteria Bacillus anthracis, they can enter the human body from the animals. Same goes for rabies. So for rabies and salmonellosis, you must have heard about these diseases. So rabies is caused by a virus, rabies virus, and there are certain animals which can get these viral infections. So mostly domesticated animals, dogs, and then rabbits. So all these monkeys, so they can carry these rabies viruses. Okay, and once a rabid dog bites a human, that virus can get transmitted through the saliva of the dog to the human from the site of the bite. And these viruses, they infect the brain or nervous system tissue in the infected, like of the infected animals. That's why the dogs that are infected by rabies, they actually are very aggressive, and because their nervous system tissues are infected by these viruses, so they become very aggressive, and they tend to bite other uh, people and other animals, because biting will then transmit that disease. Okay, and if a human gets bitten by a rabid animal, they should get the vaccine for the rabies immediately. Otherwise, humans can also get infected by these viruses, and then our nervous system tissues will also be affected. Okay, and that is why in case of rabies, it is very important uh, to note where the bite has been uh, made in the body. So, if a rabid animal bites me on my leg, okay, for the virus to for the virus to travel from my leg to my brain will take more time as compared to a rabid animal biting humans on their head or on their face. So that's why if the bite has been made on the upper portion of the body, generally the person is immediately administered with the vaccines, and the person is under more risk of having the rabies infection. as compared to the person who has been bitten on the leg. Okay. Then salmonellosis is also caused by a common bacteria called salmonella. So salmonella, it's a group of bacteria and they can cause gastrointestinal illness, so stomach issues. So and these salmonella bacteria, they generally reside in all these poultry food, so chicken, meat, fish, all these food contains these bacteria salmonella. That's why we are supposed to cook the food properly in order to kill these bacteria and wash the meat properly before cooking to get rid of this infection. This also gets transmitted from animals. Okay. And malaria I've already explained. Is this clear?
Could you please throw us the previous question slide?
Yeah. The answer would be malaria. I've written, I have to edit the slide.
Thank you, ma'am.
Yeah. Moving on to the next question. Sandflies spread the deadly disease which is known as which is known in India as Kala Azar. Which microbe caused this disease? Options are Leishmania protozoan, Ike virus, Yersinia pestis bacteria, and Plasmodium parasites. Sandflies spread the deadly disease which is in India known as Kala Azar. Which microbe caused these disease? All of you write your answers in the chat box. I can see 4 people have already answered. Anyone else wants to write the answer? Okay, so 4 of you have answered. You are saying option A, Leishmania protozoan. Let's see the correct answer. That is the correct answer. Leishmania protozoan is the microbe that causes the Kala Azar disease. So, visceral leishmaniasis, also known as Kala Azar, it is fatal if left untreated in over 95% of cases. It is characterized by irregular bouts of fever, weight loss, enlargement of spleen and liver, and anemia. And most cases occur in Brazil, East Africa, and India. Leishmaniasis is caused by protozoan parasite from over 20 Leishmania species. Over 90 sandfly species are known to transmit Leishmania parasite. So, sandfly, again, it's a type of insect which is the vector for these Leishmania parasite. So, Leishmania is carried by the sandfly, and when the sandfly bites the human population, the protozoa is transmitted into the humans. Okay, I hope that's clear. Moving to the next question. Amoebiasis is a waterborne disease. Which part of human body is most affected by this parasitic disease? Amoebiasis is a waterborne disease. What part of human body is most affected by this parasitic disease? Options are option A eyes, option B ears, option C urinary tract, and option D colon. Which part is affected by this amoebiasis disease? eyes, ears, urinary tract, or colon? Write your answers in the chat box. I have only two answers in the chat box. What about others? One answer is option D, one answer is option C. Others also answer. Okay, four of you have answered. Now all of you have answered option D. Anyone else wants to write the answer? Okay, let's see the correct answer. You all are saying option D, colon. That is the correct answer. So, amoebiasis is a parasitic infection of the colon or intestines caused by the protozoan Entamoeba histolytica or E. histolytica. The symptoms of amoebiasis include loose stool, abdominal cramps, stomach pain. Basically, it causes gastric issues. However, most people with amoebiasis won't experience significant symptoms. In some cases, it invades the colon wall, causing colitis, acute dysentery, or long term chronic diarrhea. The infection can also spread through the bloodstream of to the liver. Okay, so eventually the infection can lead to or cause liver infection, but initially it infects the colon or the gut uh, digestive tract of the humans, where it can cause gastrointestinal issues. Okay, that's clear. Moving to the next question, relative ability of a parasite to cause disease is called, sorry, the relative ability of a parasite to cause disease is called option A, vector, option B, virulence, option C, virality, and option D, viral ability. Relative ability of a parasite to cause disease is called vector, virulence, virality, viral ability. All of you write your answers in the chat box. I can see only three answers. What about others? If you know the answer, please write it in the chat box so that I'll also understand if you are understanding or not. What is the relative ability of a parasite to cause disease called? Okay, let's see the correct answer. So, three of you are saying option B, virulence. That is the correct answer. So, virulence is the pathogens or microorganisms' ability to cause disease or cause damage to a host. So, basically, one parasite or one microorganism, if it is causing infection, if there are two different microorganism and both of them cause infection, but one of them cause more severe infection than the other, then uh, we'll call the first microorganism that is causing the more serious infection more virulent than the other microorganism. Okay, so virulence is a measure of how serious a disease can be caused by particular microorganism. Okay, I hope that's clear. Let me know if you have any doubt or any questions. Okay, moving on to the next question, question number 12. Microbe human interaction can be neutral, mutually beneficial, or pathogenic. In the following list of microbes, identify the mutually beneficial microbes out of the pathogenic ones. Microbe human interaction can be neutral or mutually beneficial or pathogenic. In the following list of microbes, identify the mutually beneficial microbe out of the pathogenic ones. All of you write your answers on the chat box. Which of the following microbes identify among the following microbes identify the mutually beneficial microbes? Option A, Escherichia coli. Option B, Mycobacterium tuberculosis. Option C, Clostridium botulinum. And option D, Staphylococcus aureus. What would be the correct answer? Which bacterium is mutually beneficial for humans? Okay, I see four of you have answered option A, Escherichia coli. Anyone else wants to answer? The correct answer is option A, Escherichia coli or E. coli. It is a type of bacteria that normally lives in the intestine of all warm blooded animals. So, e- so what do we mean by mutually beneficial microbe? Mutually beneficial means it is both like the bacteria and the humans both are benefited from each other. So how the bacteria is benefited? Bacteria is benefited as it is getting a place to live, so it resides in the intestine and you thrives on the nutrients that are present in the intestine. So it is growing in the intestine of the human, so it is benefited. And how it is benefiting the humans? It helps with the digestion in the gut, so it's a gut microbe. And there are multiple gut microorganisms that are present that helps in the digestion of our food. So one of the gut microbe is E. coli. So it helps humans by food digestion, and it also prevents pathogenic bacteria from colonizing the gut. So gut microbiota is very important. All the other options that are given except E. coli, all of them are pathogenic. So Mycobacterium tuberculosis, they causes tuberculosis. All of you know that it's serious disease that is caused, it's an infection caused in lungs. Then comes Clostridium botulinum, which produces the toxin, botulinum toxin. And I had told you in the previous one of the previous session about Botox. So the Botox that is taken by certain people for preventing the aging basically is the injection of the botulinum toxin in a very small dose. So botulinum toxin can cause nerve paralysis. So when injected in very small dose at the areas where we are prone to get wrinkles, if we inject this toxin in a very small dose, the nerves around that muscle will get paralyzed, and thus the skin will not start wrinkling. The skin, basically the muscles there will get arrested, they will be paralyzed, so they will not grow further, and wrinkles will not form. Then Staphylococcus aureus, again, it's a very common disease, so it causes staph infection. Mostly these bacteria are present in our skin, and it can cause the acne. So there are certain type of bacterial infection that happens in the skin that leads to acne. Most common bacteria amongst them is Staphylococcus aureus. Okay, so all the three of them are pathogenic. E. coli is not pathogenic; it is mutually beneficial uh, microorganism. Is that clear? If you have any doubt, please let me know. Okay. Moving on to the next question, question number 13. Organisms that can make their own energy using light and carbon dioxide via the process of photosynthesis are called: option A, photoautotrophs; option B, chemoautotrophs; option C, photoheterotrophs; and option D, chemoheterotrophs. Organisms that can make their own energy using light and carbon dioxide via the process of photosynthesis are called photoautotrophs, chemoautotrophs, photoheterotrophs, chemoheterotrophs. Write your answers in the chat box. All of you write your answers. Whoever knows the answer, please write it in the chat box. Organism that can make their own energy using light and carbon dioxide via process of photosynthesis. I can see three of you have answered option A, photoautotrophs. Let's see the correct answer. Photoautotrophs is the correct answer. They are organisms that can make their own energy using light and carbon dioxide via process of photosynthesis. So in one of the previous session, I have already shown this chart. So I'll go through this again. So all organisms, they're divided into two categories: autotrophs and heterotrophs, depending on what type of carbon source they are using. So if they can directly fix the carbon dioxide, they can directly uh, use the carbon dioxide as the carbon source, that is the inorganic carbon, basically. Those organisms are called autotrophs. And if they are using organic carbon as their carbon source, so if we are like humans, we eat uh, organic carbons as our food to get the energy, right? So if you remember from the previous class, we talked about glycolysis. So glycolysis, basically glucose was broken down into energy. So that is heterotrophs. Whereas plants, they can use carbon dioxide directly and make their own food, so they are autotrophs. Then these are further divided into type of energy they are the organism is using. If the source of energy is light, then it is photo. If the source of energy is chemical, then it is chemo. So if it is any organic energy source, then it is chemoautotrophs. If it is a light source, then it is photoautotrophs. Same goes for heterotrophs. We have photoheterotrophs and chemoheterotrophs. Okay. So here in the question, it was using light, so it was photo, and using carbon dioxide, so it was autotrophs, so photoautotrophs. Okay, if it was using carbon dioxide to make chemical thing, then it would be chemoautotrophs. If it is using organic carbon source as the primary carbons, then it would be heterotroph. And if it is using light as energy source, it would be photoheterotroph. If it is using chemical energy as uh, the energy source, then it would be chemoheterotroph. So all humans come under this chemoheterotroph category, because we use organic carbon source and we use chemical energy to break down those organic carbon source into energy. Okay. Okay. Next question is also based on the previous table that I just described. So all of you should be able to answer this. Organisms that are able to synthesize their own organic molecule from the fixation of carbon dioxide and are able to produce their own source of food or energy are called. So they are able to synthesize their own organic molecules from the carbon dioxide fixation and produce their own source of food and energy. What would we call them? I just explained the whole chart, so you should be able to answer this. I have only one answer in the chat box. What about others? All of you write your answers in the chat box. Okay, I can see four answers in the chat box. All of them are option B, chemoautotrophs. That is the correct answer. So I have already explained the chart. So now you understand they obtain their energy from chemical reactions. So they are making organic molecules through chemical reaction, and they're using carbon dioxide as the primary carbon source. So they are autotrophs, and since chemical reaction is happening, it is chemoautotroph. Okay, is this clear? I hope it is clear to everyone. If you have any doubt, please let me know. Okay, moving on to the next question. What is the maximum absorption range of bacteriochlorophyll A? What is the maximum absorption range of bacteriochlorophyll A? Options are: option A, between 600 to 725 nanometer; option B, between 625 to 700 nanometer; option C, between 800 to 925 nanometer; and option D, between 825 to 900 nanometer. What is the maximum absorption range of bacteriochlorophyll A? Maximum absorption range of bacteriochlorophyll A. All of you write your answers in the chat box. What do you think would be the correct answer? Maximum absorption range of bacteriochlorophyll A. 600 to 725, 625 to 700, 800 to 925, and 825 to 900 nanometer. Which of the following is the correct answer? I can see two answers in the chat box. What about others? What would be the correct answer? Anyone else wants to write the answer in the chat box? I have two answers. Both of them are option C, between 800 to 925 nanometer. Let's see the correct answer. The correct answer is option C, maximum absorption range of bacteriochlorophyll. It is between 800 to 925 nanometer. That is correct. So both of you were correct. Moving to the next question. Membrane bound pigments that participate in the photosynthesis and photoprotection for the organisms are called. Membrane bound pigments that participate in the photosynthesis and photoprotection for the organisms are called option A carotenoids, option B carcinogens, option C phycobilins, and option D phytonadione. Membrane bound pigments that participate in photosynthesis and photoprotection for the organisms are called carotenoids, carcinogens, phycobilins, and phytonadione. All of you write your answers in the chat box. Which of the following are the membrane bound pigments that participate in photosynthesis and photoprotection? Carotenoids, carcinogens, phycobilins, phytonadione. I can see three answers in the chat box. What about others? Anyone else wants to write the answer? If you know the answer, please write. Okay, three of you. Okay, I have one more answer. So, four of you have answered option A, carotenoids. Let's see the correct answer. Option A, carotenoids is the correct answer. So, carotenoids are class of more than 750 naturally occurring pigments synthesized by plants, algae, and photosynthetic bacteria. These richly colored molecules are the sources of yellow, orange, and red colors of many plants. So, you must have seen plants with red leaves naturally present, and they have red colored leaves, so they have more amount of carotenoids than chlorophyll. So that's why generally the green color of the plant is due to chlorophyll, right? So if the plant has more carotenoid pigment than the chlorophyll pigment, they will appear as red or yellow or orangish. Okay. Carotenoids are ubiquitous and essential pigments in photosynthesis. They absorb in the blue green region of the solar spectrum and transfer the absorbed energy to the chlorophylls or bacteriochlorophylls, and so expand the wavelength range that is able to drive photosynthesis. So all these molecules, carotenoids, chlorophylls, all these pigments, they are basically chromophores. They can absorb lights at different wavelengths. Okay, so now why plants and all these photosynthetic organisms they have different pigments is to expand the wavelength range. So now if only one pigment is present, that means there's only a small wavelength range that can be absorbed by those plants. So their ability to survive in a environment with wide range of wavelength of light available, their ability to survive would be lesser. Okay, when these organisms have multiple pigments that can absorb the uh, different wavelengths of light, that can absorb different range of wavelengths, that can actually help the plant to drive photosynthesis faster. Okay, so carotenoids is one such pigment, and it is a membrane bound pigment. And these can basically absorb in the blue green region, and then they will transfer the absorbed energy to chlorophyll, and the rest of the photosynthesis process will occur via chlorophyll. Okay, is this clear? Anyone has any question or any doubt? Please let me know if you have any doubt or any question. Okay, moving on to the next question then. Water soluble pigments are not attached to the membranes are known as. Water soluble pigments not attached to the membrane. So in the previous question we talked about membrane bound pigment. Now in this question I'm asking water soluble pigments that are not attached to membrane. They are known as, again options are carotenoids, carcinogens, phycobilins, phytonadione. Write your answers in the chat box. Which pigment is water soluble, that is not attached to membrane? All of you write the answers. Carotenoids, carcinogens, phycobilins, phytonadione. I can see three of you have answered option C, phycobilins. Let's see the correct answer. That is the correct answer. So phycobilins, again like carotenoids, these are also pigment cells. These are also chromophores. They can absorb light at different wavelength, at a different range of wavelength. So phycobilins are light capturing bilins found in cyanobacteria and in the chloroplast of red algae, glaucophytes, and some cryptomonads. So unlike carotenoids, phycobilins are not present in all organisms. Carotenoid is more ubiquitous in that sense. So phycobilins are only present in these many organisms. Most of their molecules consist of chromophore, I already explained, which makes them colored. They are unique among the photosynthetic pigments in that they are bonded to certain water soluble proteins known as phycobiliproteins. So they are bonded to these water soluble phycobiliproteins. That's why they are water soluble. They are not anchored to the membranes. Phycobiliproteins then pass the light energy to chlorophylls for photosynthesis. Phycobilins are especially efficient at absorbing the red, orange, yellow, and green light wavelengths that are not well absorbed by chlorophyll A. So you had seen the absorbance range of bacteriochlorophyll A in one of the questions, right? So phycobilins can absorb the red, orange, yellow, and green light, uh, which is not absorbed by these chlorophyll A. So organisms growing in shallow water tends to contain phycobilins that can capture the yellow or red light, while those at greater depths often contain more of the phycobilins that can capture the green light, which is relatively more abundant. Now, the organisms that are living in the deep sea, at the very bottom of the deep sea, their natural light will not enter, right? But there are the light of wavelength that is in the green region, that can still reach those part. So you need a pigment that can absorb that particular wavelength. So phycobilin is one such wavelength, and phycobilins fluoresce at a particular wavelength. So they are therefore often used in research as chemical tags. So they have a fluorescence property. Basically, fluorescence means they can absorb a particular wavelength and emit another wavelength, emit a higher wavelength. So that property is called fluorescence, and these molecules have that property. So they are also used in research. They are used as chemical tags for binding, by binding the phycobilin proteins to antibodies in a technique known as immunofluorescence. Is that clear? Any question regarding this? If you have any doubt or any question, please let me know. Okay, moving on to the next question. Number of electrons required for the nitrification of ammonium are option A 4, option B 6, option C 8, and option D 10. From ammonium to nitrate, nitrification of ammonia, that means ammonium to nitrite to nitrate. How many electrons are required for this process? 4, 6, 8, or 10? Write your answers in the chat box. Okay, I can see three of you have answered option C. What about others? Anyone else wants to write the answer? Number of electrons required for the nitrification of ammonia. Okay, three of you are saying option C 8. That is the correct answer. So, nitrification is the process by which ammonia or ammonium ion is converted to nitrate. Nitrification is the net result of two distinct processes. First is oxidation of ammonium to nitrite by nitrosifying or ammonia oxidizing bacteria, and then oxidation of nitrite to nitrate by nitrite oxidizing bacteria. So we have already discussed about nitrogen cycle. So at that time I explained how the nitrogen uh, is how the ammonium is oxidized into nitrite and nitrate, and then denitrification, what is denitrification process, what is nitrogen fixation, and what bacteria carries which process. I have already discussed that. So ammonification, so nitrification of ammonia basically means the oxidation of ammonia into nitrite first, and then further oxidation into nitrate. Okay, so the number of electrons that are required for the whole process, how will we calculate? So the this is the first reaction that is happening, ammonia to nitrite. Okay, so how do we know how many electrons are involved in this? So we'll have to first balance this equation. For balancing, first we'll balance the oxygen. So here we have NH4 plus and here we have NO2 minus, right? Now, how many oxygens are there on the right hand side? on the right hand side we have 2 oxygen, and on the left hand side we don't have any oxygen. First we'll balance the oxygen. So one water molecule has one oxygen, so we'll add 2 water molecules. So on the right hand side and left hand side we have 2 oxygens. Now we'll balance the hydrogen. So we have 4 hydrogen from NH4, and now 2 water molecules will give 4 more hydrogen, so total 8 hydrogens. So we'll add 8 protons on the right hand side to balance the 8 hydrogens on the left hand side. Now we'll finally balance the charge. So on the left hand side we have +1 charge, and on the right hand side we have +8 charge from the protons and -1 charge from the nitrite. So 8 minus 1 is 7. So +7 charge on the right hand side and +1 charge on the left hand side. Okay. Now if I have to balance the charges, we need to subtract uh, 6 from the right hand side, right hand side has +7. So if we subtract 6, we'll get +1, same as the left hand side. So we'll add 6 electrons, because one electron has one negative charge, so 6 electrons will give 6 negative charge. So 7 minus 6 would be +1 on the right hand side, same as the left hand side. So that means for the oxidation of ammonia into nitrite, it is taking 6 electrons. This part is clear? Till this point, I hope that is clear to everyone. After this, the second step is nitrite to nitrate, that is NO2 minus to NO3 minus. So again, same process we will go through. So I haven't written the steps separately. So NO2 minus to NO3 minus, first we will balance the oxygen. So we have one extra oxygen on the right hand side, so we will add one water molecule on the left hand side. Then once we add one water molecule on the left hand side, we have 2 hydrogen on the left hand side, so we'll add 2 protons on the right hand side. Okay. Now we'll have to balance the charge. So on the left hand side we have -1 charge, on the right hand side we have 2 plus charges from protons and 1 minus charge from nitrate. So 2 minus 1 is +1. So on the left hand side we have -1, on the right hand side we have +1. So to make +1 into -1, we need to add 2 electrons. Okay, so 2 minus 2 plus 1 would be -1. So total number of electrons required for the oxidation of nitrite into nitrate is 2. So if you add up, total number of electrons would be 6 plus 2, 8. So for oxidation of ammonium into nitrate, complete oxidation of ammonium into nitrate, so ammonia to nitrite and then nitrite to nitrate, total process is taking 8 electrons. Okay, transfer of 8 electrons are involved in the nitrification of ammonium. Is this clear to everyone?
Yes, ma'am.
Fine.
Yes, ma'am.
Okay, let's move to the next question then. Question number 19. The order of zones from top to bottom in a Vinogradsky column is: option A, aerobic water, anaerobic sediment, and then anaerobic water. Option B, aerobic water, and then anaerobic sediment. Option C, anaerobic sediment, aerobic water, and then anaerobic water. And then option D, anaerobic water, aerobic water, and then anaerobic sediment. So what would be the order of zones from top to bottom in a Vinogradsky column? All of you write your answers in the chat box from top to bottom. So Vinogradsky column is a model, experimental model of the pond ecosystem. Okay, so the natural pond ecosystem is experimentally simulated inside a lab by creating this Vinogradsky column. So now you can think what which layer should be at the topmost and which layer should be at the bottommost in a pond. So aerobic water, anaerobic sediment, anaerobic water, or aerobic water, anaerobic water, or anaerobic sediment, anaerobic sediment, aerobic water, anaerobic water, or anaerobic water, aerobic water, and anaerobic sediment. What would be the correct answer? I have two answers in the chat box. What about others? All of you write your answers in the chat box. What should be the topmost layer and what should be the bottommost layer? Think about a pond. If a pond is there in the uh, outside atmosphere, what should be the topmost layer? Should it be anaerobic or aerobic? Then what should be the next layer? Think of it like that. All of you write your answers in the chat box. Only two people know the answer. Okay, let's see the correct answer. So two of you who are saying option B, you are correct. So the first layer at the topmost layer is the aerobic water, then comes the anaerobic water, and finally the anaerobic sediment. So this is how the Vinogradsky column is set up. So it is set up to mimic the pond ecosystem. So generally, if you want to do it in your lab, what you can do, you can take a large measuring cylinder, let's say 1 liter or 2 liter measuring cylinder, then put some natural soil at the base of the cylinder, about like a thick layer of soil. You can add some additional carbon source like eggshells or newspaper, shredded newspaper. So you can add different carbon nitrogen sources to that soil, or you can take a soil which has all these nutrients and then fill the soil at the bottom layer and then top it up with water. Okay, normal tap water, you can top it up with water and leave the whole column in the sunlight, cover it with foil and make holes on the top, or cover it with a mesh so that oxygen can penetrate, and we are using a glass measuring cylinder so that light can enter, and then leave the column in the sunlight. So after keeping the column still in the sunlight, it will start mimicking a pond ecosystem. So pond is also basically a pool of water which has different carbon and nitrogen sources, and it also can form different layers like the layers formed in this column, in this experimental column. So what will happen when you incubate this cylinder in, or this column in the sunlight? Light will penetrate up to certain point and oxygen will also penetrate up to certain point of this column. It oxygen cannot diffuse up to the bottommost layer of the column because it's a tall column, so oxygen diffusion rate is not that great, so it won't reach the bottommost layer of the column. So the region where up to which the oxygen can penetrate, that region will become the aerobic water. Below that, the region, the water region where there will be no oxygen, that region will become anaerobic water, and then comes the soil part, which will be the anaerobic sediment, because no oxygen can penetrate in this area. Now depending on these regions, aerobic water, anaerobic water, and anaerobic sediment, different microorganisms will start growing and they will acquire the regions based on their metabolic capacities. So on in the aerobic water, we'll have cyanobacteria and sheathid bacteria which can survive in presence of oxygen and which can use light as the energy source, because light can also penetrate in the top part of the column. In the middle part, anaerobic water, we'll have certain purple non-sulfur bacteria which cannot survive, which cannot use oxygen as the terminal electron acceptor, so they will use sulfur. So aerobic zone will have chemoautotrophs, cyanobacteria, which is oxygenic photosynthetic bacteria, sulfur oxidizing bacteria, which are anoxygenic. Sulfur oxidizing bacteria will come in the anaerobic zone. So they are anoxygenic photosynthesis bacteria because some amount of light will penetrate at the top part of the anaerobic water. So there, the anoxygenic photosynthetic bacteria will survive, that can use H2S as the energy source. So instead of H2O, H2S will be there in the electron transport chain. Then comes the uh, photoheterotrophs, that are purple non-sulfur bacteria, which can use light as the energy source and organic carbon as the carbon source. So they will use the organic carbon from the soil as the carbon source, and the light that is penetrating in the anaerobic region as the energy source. Then we'll have anaerobic photosynthetic purple and green sulfur bacteria, and then anaerobic respiration will occur in this region. Then we'll have, below this we'll have sulfur reducing bacteria, which can use sulfur and reduce sulfur into sulfates. So sulfur and sulfates will be reduced. And then lastly, we'll have heterotrophic fermentation occurring, where sulfates are reduced to sulfides. So H2S will be formed, and that H2S will rise up. H2S is gaseous, so they can rise up. So all the bacteria here, the the photosynthetic sulfur bacteria, basically, they will release sulfates, and these sulfates will be used up by the anaerobic uh, bacteria that are residing in the anaerobic sediment, and they will release H2S, which will be used as the terminal electron acceptor by the photosynthetic anaerobic bacteria or anoxygenic bacteria. Okay, so a whole ecosystem is developed in a column. So this is a classic experiment developed by Vinogradsky, and it is performed in many microbiology experimental labs. Anybody has any doubt in this? I hope that's clear. Moving to the next question. Photosynthesis occurs in, options are chloroplast, Golgi body, endoplasmic reticulum, and nucleus. Photosynthesis occurs in chloroplast, Golgi body, endoplasmic reticulum, and nucleus. What would be the correct answer? Please write your answer in the chat box. Photosynthesis occurs in chloroplast, Golgi body, endoplasmic reticulum, and nucleus. I can see three answers in the chat box. All of them are option A, chloroplast. Let's see the correct answer. Chloroplast is the correct answer. Photosynthesis occurs in the chloroplast. So chloroplasts are the organelles that carry the chlorophyll, pigment chlorophyll, that can uh, absorb light and carry out the photosynthesis process. Okay. Next question is, final product of Calvin cycle is option A, RUBP, option B, glucose, option C, dihydroxyacetone phosphate, and option D, glyceraldehyde 3-phosphate. Final product of Calvin cycle is RUBP, glucose, dihydroxyacetone phosphate, or glyceraldehyde 3-phosphate. What would be the correct answer? Write your answers in the chat box. All of you write your answers in the chat box. Final product of Calvin cycle. RUBP, glucose, dihydroxyacetone phosphate, and glyceraldehyde 3 phosphate. I can already see 4 answers in the chat box. All of them are option D, glyceraldehyde 3 phosphate. That is the correct answer. So, Calvin cycle is generally present in plants where they use the carbon dioxide, they fix the carbon dioxide and ultimately produce ATP. So, in the previous session, we saw the glycolysis pathway and TCA cycle, which were eventually leading to the aerobic respiration process and the ATP generation, right? So, any process that any biological or biochemical process that is happening inside the body or inside any organism is basically ultimately to generate energy. In plants, they can directly use the carbon dioxide because they are photoautotrophs, we have already seen, right? So, what they do, they use the carbon dioxide to make the ATP. In humans, what was happening, they were using the sugar or carbohydrate sources to break down into simpler carbon molecules and then ultimately generating ATP. So in plants, Calvin cycle occurs where CO2 enters the cycle. 3 rounds of CO2, 3 molecules of CO2 will enter the cycle one by one. So 3 round of this cycle will lead to one molecule of glyceraldehyde 3 phosphate. So one carbon dioxide will enter and go through this cycle, then another will enter, then the third one will enter, and finally we'll have one glyceraldehyde 3 phosphate as a output. And you know the glycolysis pathway was generating this glyceraldehyde 3 phosphate, right? So there's another process called gluconeogenesis, which is the opposite pathway of glycolysis, okay? And by that process, glyceraldehyde 3 phosphate can actually uh, form glucose again. So we had seen the opposite process happening, glucose to glyceraldehyde 3 phosphate. Now in this process, glyceraldehyde 3 phosphate will form glucose. So if you remember, in the glycolysis, one molecule of glucose was generating 2 molecules of glyceraldehyde 3 phosphate, right? So here also we need 2 molecules of glyceraldehyde 3 phosphate to make one molecule of glucose. So that means 3 cycles of this pathway generates one molecule of glyceraldehyde 3 phosphate. That means 6 rounds of this cycle will lead to 2 molecules of glyceraldehyde 3 phosphate, ultimately producing one molecule of glucose. Now you can see the total cycle, if it turns 6 times for the production of one molecule of glucose, it will require 6 molecules of CO2, 18 molecules of ATP, and 12 molecules of NADPH. Okay, so it will require 18 molecules of ATP and 12 molecules of NADPH. Later, these glucose can be broken down by the process and these ATP will be restored. Okay, plants get their energy from light, right? So the energy used for these processes, they are ultimately sourced from the light energy, the sun, basically sunlight. Okay, is this clear? So the final product of Calvin cycle is glyceraldehyde 3 phosphate. Is that clear? Moving on to the next question. Reduction of sulfates and sulfides to hydrogen sulfide is done by which group of bacteria? So we had already discussed the Vinogradsky column, so you should be able to answer. Reduction of sulfates and sulfides to hydrogen sulfide is done by which group of bacteria? Option A, aerobic sulfate reducing bacteria. Option B, photosynthetic sulfur bacteria. Option C, anaerobic sulfate reducing bacteria. And option D, heterotrophic bacteria. All of you write your answers in the chat box. I just explained the whole Vinogradsky column, so you should remember which part or which group of bacteria was carrying the reduction of sulfates and sulfides to hydrogen sulfide. Aerobic sulfate reducing bacteria, photosynthetic sulfur bacteria, anaerobic sulfate reducing bacteria, heterotrophic bacteria. I have two answers in the chat box. Anyone else wants to write the answer? All of you who know the answer should write the answers in the chat box. Okay. Three of you are saying option C, anaerobic sulfate reducing bacteria. That is the correct answer. So I have already explained in that Vinogradsky column. At the bottommost layer in the anaerobic sediment, these anaerobic sulfate reducing bacteria will reduce sulfates and sulfides into hydrogen sulfide, which will be used by as a terminal electron acceptor by the photosynthetic anoxygenic bacteria at the middle layer. Okay, so this was the last question of the session. If you have any doubt or any question regarding any of the topics of the session, or if you have any doubt in any of the questions discussed today or related to your assignment, please let me know. Yes, you may ask what doubt you have. Fill in the blank. Limiting nutrient can be. Just a second. I think the answer should be micronutrient. Okay, let me check and get back. So limiting nutrient basically means any nutrient that is present in the limiting amount, so that will get depleted first. I According to me, the answer should be micronutrients. I'll look into this and I'll get back to you in the next class. It should not be only macronutrients, either it will be B or C. I'll just give me some time. I'll In the next class I'll clarify this doubt. Let me see. Okay, we'll have the next class this Tuesday, so there I'll let you know. Okay, any other doubt? Did you get the answer key for the previous assignment clarified? You had a doubt in the question from week 10 or week 9, right? Week 9, I think. Was that clear? Did you mail the team as I asked? Yes, you can mail them. If there are many errors in the answer key, if there are errors, you can mail them. Tell them that this should be the correct answer, so they can also have a look. For this question, I'll just confirm the answer in the next class. Let me read into it. I don't think it should be only macronutrients, but I don't want to give you a wrong answer, so let me confirm and then get back to you. For the week 8 and 6, for that radioactivity question, for week 8 question, I think you should mail them. Okay, sure. Okay, then, no other questions today. Anyone else wants to ask anything? Okay, if there are no more questions, then we can end this session. So, thank you all for joining today, and we'll see you next Tuesday.
Thank you, ma'am.
Yeah, thank you. Bye bye.